Hi. In this video, I'm going to go through a exploration of the GDC in order to create sets of data that has sample cases grouped into little, what we call cohorts, in order to allow for downstream analyses where you are taking one group of samples that are very similar to a second group of samples, but differ in some sort of key, likely biologically relevant way. And then you're comparing their differential expression, or well, their gene expression, and then seeing if it is differential through various statistical analyses. In my class, using the program DESeq2. However, in order to do that, I first need to identify what groups the different cases of data should be in. Additionally, if I don't even really know what my comparison is going to be, I need to kind of go in with the scientific question and try to resolve that. So in this particular example, I'm going to go in trying to explore something related to a really common, likely inherited mutation. Now, I already know going into this, I want to look at the CRAS gene in particular, and that's partially because I've already gone through and seen like, well, what genes have lots of copies of the same variant in this data, which generally is going to mean that what mutations just exist in the natural population that tend to be correlated with cancer, since this is primarily cancer data. So with that, let's start. Let me move me. Hi, I'm in the corner now. All right. So to start off, we're going to go with cohort builder. And in order to do an analysis, you have to make sure the data is there. Now, in my example, I'm about to use 3 separate pieces of data. I'm going to use the count data. That's what differential expression analysis will be occurring on. In my class, all of you need to include this as a filter. I'm going to include the TCGA as a filter. And that's because, again, for my class, we're using TCGA data, not the other projects, which sometimes have their own caveats that I don't want to have to deal with. And finally, open access data. So those are going to be the 3 filters everyone in my class has to use. But then I'm also going to be considering 3 other similar analyses at the same time. And that is mutation screening of simple somatic mutations, so like mutations within non-sex cells. And so I can't prove that these are heritable, although what we're going to do is look at gene frequencies across the GDC for the same mutation over and over. And since the genome is enormous, seeing the same mutation over and over generally is going to mean that that is an inherited mutation, not something that just naturally occurs. There's probably some exceptions to that, maybe some places within the genome that just tend to mutate due to their structure. But for the most part, that's going to mean an inherited mutation if it is the same mutation in many different people. Finally, we're going to include copy number variant screening. And so a common issue with cancer is that because of disruptions to the DNA replication process due to the unhealthiness of the cancer cell, they tend to accumulate mutations that just kind of leave out large chunks of the genes. So maybe like an entire arm of a chromosome or a part of an arm of a chromosome. And when you do that, you lose copies of genes. And if those genes that are there are genes intended to suppress the formation of tumors, that's a problem. On the other hand, sometimes you'll get duplication of chromosomes. And again, if you have a gene that its job is to be produced at a very fine level of scale, and all of a sudden you have multiple copies, well, that will actually correlate with an increase of expression, and that might cause an increase in the amount of cell division that it does, and thus again leading to cancer. So copy number variants, very, very important for cancer research. That's really the level I understand these. And so for my experiment, I'm wanting to look at this presence of this mutation versus, well, the best comparison is no mutation in that gene. Please note, I didn't say not that mutation. There is a difference. We're going to actually have to do a lot of work to make that happen. So has the mutation, has no mutation. And so as we do that, we're going to have to remove everything that has other mutations, including copy number variants. So that's my first step. We'll revisit this process, but that's my first step. Isolate the cases that belong to the sets of data I'm interested in. So let's start off with TCGA because class stuff. Available data. It needs to be open access. You're not allowed to use the controlled access data unless you get special permissions from the government, I guess. But we don't have those for this class. That would take a lot of effort and wouldn't really be all that useful. So, and I probably couldn't get it. So the first thing we need is gene expression quantification. I can actually see once I click on that, with those two combined, that leaves us with only one particular type of gene expression quantification that is just generally available to the public, and that is the star counts. So this will become one of my cohorts, is what we call this. I haven't saved any yet, so I'm just going to click save, and this is going to be TCGA open counts. So one of my sets of data. I'm just going to tell you, there are two other sets of data I need. I need that mutation screening. Ooh, I clicked on gene expression, but it turns out star counts is enough to reduce the data types to only a single type. So I'm going to have to remove that one as well. I could even click it up here if I can't find it down below. I will tell you, I did some work behind the scenes. I can tell you that those simple browsable mutations are termed the data type masked somatic mutations. So this is going to tell us do they have a mutation in each gene? From this, I can go through and click these single things, but just masked somatic mutations and open access, that's enough to get me down to a single analysis. And I'm going to save that as a cohort. So no longer counts, this time SSM. Done. And finally, for my analysis, I want to remove copy number variants because I want this to be very, very specific. And so again, I just kind of had to do some research behind the scenes, figure out what is the copy number variants that we can typically see within the mutation screening, and it turns out it is this gene level copy number. And if you actually went through here, you'd see there are a few other things that kind of sound like they might be. And yeah, they might have been. I had to kind of click through and do some research first. Within this, there are multiple types of copy number variant. We're not actually going to be using this data outside of using it to filter the cases. And so I'm just going to leave these as is because I'm just going to want to pull that data from wherever it's coming from. So with that, I'm going to save this. CNVs. And it just for the record, it may turn out that this isn't perfect at removing CNVs. We're going to find out, but let's start analyzing more. So set operations is going to allow us to compare sets of data via various do they intersect or do they where do they, is it one or the other, things like that. So in this view, we're seeing that per circle, which corresponds to one cohort, we're seeing a numerical breakdown of how they intersect in part. So 703 belong to 1 and 2, but not 3. So that means 703 have copy number variant data. They have count data, but they don't have open access mutation data. 9,614, though, have all 3. So I'm just going to click on that. Okay. TCGA open, CNVs, counts, SSMs. Cool. So that's going to be kind of my starting point for exploring this GDC further. Now, at this point, what I'm going to do is I'm going to make that my current cohort. My whole idea for this project was I wanted to look at inherited mutations that were very frequently found within the database. And so if I come to this gene mutation frequency, there's two main tabs. We have genes, we have mutations. genes is each gene ranked by the total number of cases across my cohort. So right now, the most common most common mutated gene, any mutation, is TP53. But TP53 is usually a gene that is mutated throughout one's lifetime. That's what leads to the tumor suppression failing as TP53, whose job it is to repress the formation of tumors, becomes mutated, cancer starts to proliferate. So since this is a bunch of data describing tumors, TP53 is very commonly mutated. However, since that's an acquired mutation, and TP53 is a fairly large gene, many of the mutations are completely unique, at least within this data set, if not within historical times. But they're not usually inherited. To see inherited mutations, I usually will need to go see mutations that are common just in general. And so like I can see the most common mutation in the entirety of my open access CNVs, counts, and SSMs is this particular mutation in BRAF. With 547 of my 9,614, so a full 5.7%, that's enormous, of people who develop cancer inherited this mutation, at least within the scope of the GDC, which does have a lot of biases that are like, it's a lot of white men, for the record. Like, it's just a thing to be aware of. So, if you have a mutation that's common within European ancestry, that's going to be really common within this data set because of those inherent biases. There are other biases as well. These are people who agreed to have their data within this project, and that does its own self-selection. So you're not going to find people who like reject modern medicine within this. So you're going to have trouble finding certain subsets of the population, or people who just don't exist in the United States at a very high frequency, and therefore weren't included in this analysis either. So there's just some inherent biases for a number of reasons. If you want to see across everything within the GDC, you can click this little drop-down menu and it will break it down, not just within our cohort, but within the GDC at large. So we can see that this particular mutation, very, very common within, I think that's thyroid cancer, 57%, absolutely wild. Very, very common. In fact, the majority of people who develop thyroid cancer seem to have this particular gene, or at least the majority of people who then got inducted into this analysis. Maybe there is a selection there. I'm not sure. Anyways, that's the idea. That's what we're visualizing here. So I kind of basically had browsed through this. I selected this particular mutation, and that's because while it is present within groups like the PAAD, the pancreatic cancer, it's not the entirety. So it's at a high frequency, but it's at not such a high frequency that I can't find anything to compare it to. And yes, this took some practice. It meant that I went down some bad roads that didn't quite work out. But that said, I'm going to select these. So there are 187 that have this particular mutation. So has specific CRAS mutation. Now, that doesn't mean that's the only mutation in CRAS they have, but they do at least have that specific CRAS mutation. So we're going to come back to that. So that's great. There's some other information I need as well, though. I don't just want a cohort of cases with a specific mutation. In order to remove cases that have any mutation and kind of create a version that has no mutations in CRAS, there's no button for that. And so I have to kind of go indirectly. So what I'm going to do is I'll eventually take my filtered data, so like this, and I will compare it to the cases that have any mutation in CRAS. So to do that, I need to create some cohorts of people who have any mutations in CRAS. So what I'll do is I'll filter my mutation data to just the CRAS gene. So I could do that here, or I could do that here. Give me one sec. I need to pause a video because my battery is dying. And we're back. So when I filtered that at the top left, or here. I created a view that shows me the CRAS mutation. Now, the CRAS mutation actually was fairly high up in that list, and so I probably could have just scrolled down and found it. But sometimes if you're working with a different gene that's not at the top, it's helpful to be able to filter. So I'm going to create CRAS SSM. So these are 732 cases that have any simple mutation in CRAS. So that's going to be things like indels and SNPs. Because I also filtered for C and V right there, I can see that of the 9,614, 2,000, so 21%, have at least a copy number gain. So that's pretty high. It seems that this gene tends to be increased overall. Very interesting. Definitely a lot of mutations in this gene. It's clearly very important to cancer development. So I'm going to say CRAS, C and V gain. Notice for the record, I don't remember if I've said this yet, but a C and V gain is going to be like a duplication event. And that's just what happens with cancer. It messes up during cell replication or manipulating the chromosomes in some way, and you can sometimes just end up creating new copies of genes. And that tends to increase the amount of copies of that gene being produced and expressed, which leads to disruptions. We also have some copy number variant losses. So those are going to be usually like large deletions that have deleted the gene large enough that it doesn't end up in SSM. And that's just because different types of analyses will be focused on different types of variants. Well, the SSM, simple somatic mutations, is inherently a simple mutation. And it can't handle something where it's like the whole arm of the chromosome has been removed. So CNV or CRAS, just to keep with my nomenclature, CNV loss. Now I will just emphasize it is good to stay organized with your names, and maybe there's a significant chunk of that that's just practice. Just for the record. Okay, so if I want to then do a subtraction to get the samples that do not have mutations in CRAS, in order to do that, I will need to do something that combines those three, because the maximum number of samples that I can include inside of a set operations is 3. Now, for the record, I could go through and I could just kind of iteratively filter my data. So just compare like open CNV counts SSMs to SSM one at a time. But instead, what I'm going to do is I'm just going to use a different strategy. I'm going to combine these in a set operations and just like make a super one. So I'm just going to have all the wedges. I picked even the ones that don't exist. So like there aren't copy number increases and losses at the same time. It's one or the other. That's why these two are 0. But when I come here, I can see these 3,200 cases that have any mutation in CRAS. Okay. So from that, I can now do that subtractive process. I can take any mutation. So I can take everything, take any mutation, and get the difference of 6,414 that have no mutation in CRAS, if this worked, and we'll see if it worked. So TCGA, we're just going to call this TCGA filtered, no CRAS mutation. So I'm going to set it as my current cohort. I'm going to go to the analysis center, just validate that that worked. I'll be honest, this is the second video I made. I don't know why it didn't work the first time. I probably just clicked something wrong. In the video, I then show that we can just kind of scroll through and we can just keep checking and we just keep working and kind of messing with the data until we're confident. But I thought it was a little bit confusing enough. Okay, so it does seem to have worked. That I can search for CRAS, nothing appears. If I want to turn this off because I just don't trust it, it still doesn't appear, even though I'm not even limiting it to the cancer gene census. So that effort worked. We have removed all cases with mutations in CRAS. So now we need to go work on actually filtering this data to just samples that are easily comparable. So I have two main things I'm looking at here. I have TCGA filtered, no CRAS mutations. I have has specific CRAS mutations. And by far the smaller one is the has the specific CRAS mutation. What I generally recommend is, because you can only get so small, like around with this type of analysis, I recommend 6 or more. Actually, for my class, I require 6 or more, but just in general, for this type of analysis, you will need a good number of samples. Ideally, quite a few, but for my class, you're limited to 6 or more. So we're going to filter this down, and we're going to see like what filters can we apply and still have data that is comparable. So let's see what projects this can be involved in. So I can see colorectal cancer, pancreatic cancer are very high. Let's just take a look at some of these. Pancreatic cancer going down to about 42 if I go to a single disease type. Within those, well, that's not great. So I can see over here underneath my head. Sorry, I'll just sink down. You can still hear me, yeah. So underneath my head, I can see that the tissue specificity is not super high. And then like I could, yes, maybe I could kind of group these. So like adjacent sections of the colon, but the colon is a big dynamic organ. There are a lot of different sections with a lot of different tissues. And I don't want my analysis secretly, like, or undesirably becoming what's the difference between cancer in the ascending colon and the cecum. I want it to be focused on this mutation. And so I didn't end up choosing this one. I ended up choosing the pancreatic cancer data set. And there was a number of reasons, but the big one being I could do some filtering, and this would make a good demonstration, because I could limit it to a single disease type. That makes sense. I should probably not have multiple diseases. I don't really know enough about these diseases to know if the adenomas and adenocarcinomas should be comparable to ductal and lobular neoplasms. They probably shouldn't. I could see that once I do that, I get down to a single primary diagnosis in a single site, and largely in a single tissue, the head of the pancreas. And I don't know a lot about the pancreas, not enough really to tell you, oh yeah, the head of the pancreas is definitely comparable to the body of the pancreas or the tail of the pancreas. This is no origin specified, so this is just some part of the pancreas. They didn't record the information finely enough. But since it's an option, I'm going to pick the head of the pancreas, and especially since I don't really know much about the pancreas, I don't know if it is comparable. So I'm going to choose to control it. Now, this one is definitely much more my experience based. If I see something like a 2-to-1 ratio between genders, that may mean that there is a significant interaction between maybe lifestyle or maybe like hormones that males and females are exposed to differently. And so I may want to select those. The primary deciding factor for me is, do I have a decently high number of cases in general? It's like if it's really small, even a small difference can have some impact. Or if there is just a large skew towards one or the other. So here, a 2-to-1, I would say that's kind of on the fence of whether or not I should remove it. However, it's enough that I'm gonna probably pick it. I will say, in my experience, race, ethnicity don't end up ever really showing up. I've had a number of students who try to do like racial comparisons. They pretty much don't pick up anything. Really, race, and maybe ethnicity, although our ethnicity scores, according to the USA Census, are just very silly. Our race descriptors are really much more based around like a very small number of visual cues, not really the genetic history of that person. So like Black or African American is going to include people who are from Africa, or which is, an enormous continent. And it's also going to include people who have been in the Americas for a number of generations because of a bunch of gross social stuff. Often will have like mixture of different genetic backgrounds. Like these are hugely diverse populations and really shouldn't be compared in like without a lot more consideration of your experimental design. And so I'm just throwing that out there. It doesn't tend to work very well in my experience. And you don't tend to get very strong signals because there isn't a lot of difference. The main thing will be where you do have a subset of the population with a specific mutation that is inherited. And when you do that, that's probably actually what you're wanting to filter upon. So like in this case, it's a little bit hard to tell, but because of like the inherent biases, I picked a specific mutation that probably has a single or at the very most a couple of common ancestors of origin. And you know, if that was a person that existed 2,000 years ago in Europe, then yeah, it's probably going to be mostly associated with white people. Although it may also have been introgressed all throughout the world. I don't know. But anyways, that's what we're kind of seeing here. I'm not going to select any of these other ones. Age, often a good one to view. You can see this down here. I do have a number of older patients in this. Age does sometimes associate with a signal, not as strong as I would probably have guessed, but it does sometimes come up. If you are doing like a severity discrepancy as your primary grouping, I often suggest you do an age at diagnosis control. Especially in anything with vital status, I don't really see a large difference. That said, it's a really hard one to control for because age obviously becomes a huge factor. So you can do it. Vital status has definitely shown some pretty interesting results before, but they had to apply a lot of filters to really make it very convincing as of a comparison. Anyways, I could keep scrolling through here. The rest of these looked pretty well controlled. A specific morphology, specific section. I've already kind of controlled for those up above, so I'm not going to select it there. Prior treatment, some of this other stuff I'm not so interested in. The pathologic stage is probably the most important one for me to include in my particular analysis. I'm going to choose 2A and 2B here. And then what I'm about to do is I am going to remove a filter. I'm going to remove this 187 case IDs. And what I should see is that when I do that, my current set of 20 cases should go up because I'll no longer be restricting it to just those that have a specific CRAS mutation. It should be any of the samples within the TCGA that meet the rest of these criteria. When I do that, I may see that this changes. So I'm not sure. Let's do that now. I think I've sufficiently done this. There are maybe some biospecimen. I could say, oh, yeah, we need to have the tumor sample. I'm wanting to compare the tumors to tumor data. It should be the primary tumor, not other things. But let's go to that stage classification. Let's hit that filter and turn that off. When I do that, I can see a bunch of other stages have popped up. And so yes, this was an appropriate filter to apply, as I can see that there are some stages of cancer I probably shouldn't be comparing those largely stage 2 cancers to. They would just have different profiles because they were at a different stage of cancer development and just stuff had started to go even crazier. If I continue to explore it, I can see that there is still only primary tumors. So while nice, I didn't have to include that one. I can see there's a number of preservation methods, although I don't really know which one to pick. And I don't really recommend choosing through these unless you need to. And we'll talk about later on. So finally, I'm going to save this. I'm going to call this just like, it's the pancreatic data that I filtered. And so, let's try that again. One sec, my keyboard turned off. All right, there we go. PAAD filtered. This is like all the cases that have matched my filters. Okay. So let's start doing some comparisons and pulling out the final sets of data. Then we will try to do some checks just to make sure we were successful. So with this, I'm going to go back to set operations. I'm going to pick my PAAD filtered. I'm going to make sure that all of those had the right set of data because I did remove some filters from that. And then I'm going to pick, okay, so actually we could do TCGA filter, no crass mutations. And then we could pick any mutation, and that'll just help us double check things. Okay, looking pretty good. So we have PAAD filtered. We have 13 that overlap with no crass mutations, but do not overlap with any mutation, which makes sense. They shouldn't. We can see 4 over here and 39 that even though they were within the PAAD filtered, they did not belong to this no crass mutation. So I don't really want them. These 39 are going to include the samples, although they won't just be limited. They're going to include the samples that we want to compare to. But for right now, I was mostly just targeting these 13. It does turn out that I could have just compared S1 to S2. I did include the third one just to help me double check things without having to do more comparisons. But this will be PAAD filtered, no crass mutation. And that should be one of my main groupings. So that one is almost done. I'll need to double check it, but before we do that, let's make the other comparison. So the mutation we're interested in, and then just that. Okay. So for that, let's do PAAD filtered, specific crass mutation. There it is. And let's just throw crass any mutation in there again, just to double check stuff. Okay, so S1 we can see is PAAD filtered, has the specific mutation is S2. Well, this 20 is going to also include has any other mutation. And so this is essentially PAAD filtered, including the specific mutation, but not just the specific mutation. In order to get just, we're going to have to do a little bit more work. So that one's not fully mature yet. And you may not have realized this if you were just kind of exploring things on your own, but that's why we always need to check it afterwards. So we're going to go to mutation frequency. We're going to select, let's just start off with the one that should have worked, no crass mutations. Okay, so filtered down to crass, no longer there. Again, if I don't trust it, I don't know why I do trust it, but I could turn off that filter and it's still not there. No crass mutations seems to have worked. PAAD filtered, including the specific mutation. In this case, all 20 and 20 have a simple somatic mutation. If I go to the mutations tab, I can see that 20 out of 20 have that specific crass mutation. If I limit that list of simple somatic mutations to crass, I can see that that is the only crass mutation that is in the SSM that they have. Now, I said that it wasn't mature yet, and that's because if I come back over here, I can see that there are actually still some with copy number variants. And so for this, if I really wanted to make a really well-controlled experiment, I should remove these as well. So I'm going to say CNV gain 2, and it went away before I pressed enter. That sometimes happens. I don't really know why. It seems to be a glitch in the current version. That's annoying. CNV gain 2 and save. Okay, it worked. Now, once that starts happening, it does tend to last for a bit, so bear with me. CNV loss 2. Dang it! That was so close. I'm sorry for shouting. Come on, come on, come on, come on, come on. Yes, got it. Cool. All right. Now we're going to do some subtraction. Pull those four up to four. If there's an overlap, we'll see. Up to four samples out of our filtered, and then we should have our mature data. So pad filtered. Actually, let's just do lots of things. There we are. Pad filtered, including the specific mutation. CNV gain 2, CNV loss 2. Okay, so with that, I get my 10 samples that do not have any other mutation. So this is pad filtered, has only specific crass mutation. And that should be my final thing. So as always, we should go double check it because this is hard stuff and it's hard to think through all the possible combinations. I can see 10 out of 10 have this mutation, 0 out of 10 have any copy number variants, and we already checked the mutations tab, but let's just do it again just for completion's sake. Boom. So we're good. Only this single mutation. At this point, now that my groups are ready to go, I'm going to go to repository. This part's very simple. Once in repository, you just need to select certain filters that let you get the gene expression. So like I can pick transcriptome profiling, I can pick gene expression quantification, but I need to pick star counts. That is the input for the pipeline we're doing in this class. I also need to pick open access. You can only use certain files. Now, I've also selected some other common things that are important. So tumor, primary tumor, as opposed to some of the other ones. All of these are going to help me just target the tissue I'm most interested in. There are sometimes other tissue types like metastatic tissue and blood tissue and things like that. Sometimes you'll also see like you'll have done all of these filters and you'll still have like 12 files for 10 cases. And then those extra 2 files will be things like other preservation methods and other samples that were taken and then sequenced. In those situations, I find the easiest way is actually just to remove them downstream after downloading the file. But you can try coming through here, and like sometimes you may see like FFPE, that's formalin and paraffin impregnated tissue. It's a type of preservation method that's pretty different from these other methods. That's usually the issue. That's probably the most common reason why there's a discrepancy here. But there may be other reasons. Sometimes just people got sequenced multiple times. Maybe one of the sequences worked well and one didn't. But in the end, mine worked well. All 10 files, or all 10 cases have exactly 1 file associated with them. So I can actually just download the sample sheet. So you can see I've already done this before. I can then repeat that process for, actually before I repeat that, I should go save that. Make sure I give it a good name. So I'm going to, I could edit it with Notepad, or I could open it with like a particular file. So like maybe Windows, Microsoft, Excel. I don't want to have to navigate to Excel on my own on the video. But I could just open it up in a text viewer. So this is Notepad++, one of my preferred ones. And then I could rename that file to be, wait, what am I looking at? I am looking at the has the mutation. So has crass mutation. Okay. I could save that, inspect it, just make sure it all looks good. I could then pull that into Excel and do some editing if needed. But the important part is that I know what group of files is what. So at that point, I would just repeat that process for the other one. Let me do that and I'll show you how to open it up in Excel. So pad filtered, no crass mutation. There we go. Okay, so that was 13 cases. That's all adding up. 806 files. Oops, need to do my filters. Normal tissue. Workflow type is where the star counts are. So really I don't need to click the rest of these because just selecting star counts is specific enough. Scrolling through here, the rest of this all looks good. Solid tissue, primary, cool. 13 files, 13 cases. I'm ready to download. So download the sample sheet again. This time I'm going to show you how to open this in Excel. So I'm going to open up an instance of Excel. This is just going to be the easier way for most students than trying to navigate the choose a program. I'm going to just open up a blank document and then browse. So I went to open, browse, and then I'm in my downloads folder. Right now, you can see it. If I had like the default settings, you probably would have only been able to see certain file types. But if I change that to all file types, it's now showing me that file. So GDC here, it pops up this message asking how were the columns delimited. This is a tab delimited file, and I can see down in my preview that it's currently working. I'm going to click finish. And when I do that, I can now see this. And there's a lot of things you may want to be looking at. The big one being like, do I have any duplicate samples? This will highlight anything that's the same sample. So for instance, if I just copy and paste that, all of a sudden it's highlighted because there are duplicates in there. I will also usually check the case ID, but the really important one is that sample ID. You can't have any duplicates there. But if everything's looking good, I'm just going to save it with a modified name, no crass mutation. There we go. And one last step I always recommend, especially if you did end up having to edit things, this is just a good tip for Excel, is just select everything. I pressed Control A to do that, then I pressed Control C to copy it. Then I opened up a new sheet and saved that. And when I do that, because a TSV file can only contain one sheet of data, that helps make sure that if like adding something here created a whole new row, I haven't created an empty row. So just a nice little data cleaning trick for you. Okay, at that point, I'm actually done and I'm ready to move on to the DEC pipeline. I'll be showing that in a new video later on. Actually, I think it'll be part of a homework, so I may just tell you to figure it out. But good luck, and let me know if you have any questions. Bye.